Okay, can you see my slide? Yeah, okay. So he's got nickname Jimmy, but the original name is Jose, right? Jose, that's pronounced. Yeah, it's because he's Spanish man originally. Yeah, so Jose, but then famous nickname is Jimmy Botella. He's a professor at Plant Biotechnology School of Agriculture and Food Sciences in University of Queensland, Australia. The education background, he did the degree in quantum chemistry from University of Madrid in Spain, in the home country, and then a PhD also in Spain in biochemistry from University of Malaga. And then he did postdoctoral as a postdoctoral fellow at Michigan State University and Pennsylvania State University in the USA. And then since 1995, he moved to Australia to join University of Queensland, and since then he's based in Australia. He's the one who founded the Plant Genetic Engineering Laboratory, PGEL. So we have the same. Our center name is Genetic Engineering Research Center, specializing in the field of tropical and subtropical agricultural biotechnology, and research and patent that he already been doing and obtained the production and field trial of world first. I put it in bold because it's world first genetically modified pineapples, and the development of ultra fast nucleic acid purification technology. It says that less than 30 seconds. It's just like, you know, things. And the PGEL, the laboratory, has also developed a number of platform technologies that can be applied to multiple crops to confer resistance to pathogens and significantly reduce the use of synthetic pesticides in crops. And he has also developed simple and extremely sensitive diagnostic technologies for crop diseases, and that can be used in the field without the need to transport samples to specialized laboratories. And he also has developed new technologies for the detection of food pathogens with important application in food safety. He has 11 international patents in the field of plant biotechnology, and he's also one of the founder of 2 biotechnology companies. Publication over 150 scientific publications, and his work has been cited in excess of over 70,600 times. The age index is 52. And over the last 20 years, collaborative research with across the countries internationally. You can name it here: USA, Germany, and in Asia, Malaysia, India, Philippines, Thailand, China, but not yet with Indonesia. So now is the time with the Indonesian Research Institute, which is with us. Okay, the award, he received a lot of awards in here. I don't want to read all one by one, but here, including the one from China, the one from Singapore, and he also received prizes and honors and awards in Australia. So for the last one is from the University of Queensland, also as an excellent teaching. Yeah, must be a good lecturer, a good teacher. Yeah, and also professional fellowship from Chinese Academy of Sciences in 2014. Okay, without further ado, I would like to hand over the laptop, the screen, to Professor Jimmy Botella. So the time is yours. So at the end, we will have discussions, but also, as Bu Ratih mentioned, not just discussion on this topic, but also you can discuss on the possibility to be his PhD students in Australia. Okay, so let me stop share here, then I will share his slide. Can you see his slide? Okay, time is yours.
Okay, thank you, everyone. Before I start, I want to thank Brim for the hospitality that for me since I got here, especially Professor Eni, because she having only 5 days here, and she made me feel like I'm at home already. So she's been not only nice, but also very helpful for everything. I had some health problems when I got here, but she took good care of me. So thank you, everyone. Today I'm going to talk. I would like to see this. Yes. Yep. Okay. About point of diagnostics. All right. This could actually have a different title. A lot of people assume that there are things like nanotechnology or molecular biology that are only good for super labs, labs that have a lot of resources. But the truth that we have discovered is that we can actually use very complicated technologies to produce a very simplified output. And that is essentially what I'm going to be talking today, this part of my lab. We do have a lot of research on genome editing and a number of other things, but this is my where I'm going to be focusing today. So when we started doing this, was mostly thinking about being able to work in places in Southeast Asia that do not have too many resources. It's what we call resource poor environments. Therefore, what we wanted to do was to develop very simple diagnostic assays that we could do it in the field with minimal equipment. There is no such a thing as no equipment. That doesn't exist. Everybody needs equipment, but we wanted to do the minimal amount possible that we could use. Okay, for this is diagnostics. For those that already know about it, this is going to be redundant, but those who are not familiar, there are a number of requirements that you need for diagnostic. One is it needs to be sensitive, meaning that you can detect the disease very early. When you get plants that are dying, it could be too late for it. It needs to be accurate, meaning that there are very similar pathogens with very similar symptoms, but you need to be able to distinguish between related pathogens. It needs to be reliable, meaning that you need to get consistent results. It needs to be fast. There are really good methods that take 2 or 3 days. That's not good enough for what we want. And it needs to be flexible, meaning that new pathogens are coming all the time. New strains of the same pathogens are coming all the time. And a typical example is TR4, tropical race 4 of Fusarium, that all of a sudden, came and then started devastating bananas. So the first thing is, what is the resource poor setting? When I talk about that, people think, okay, well, Jimmy is going to the middle of Africa in a jungle or blah, blah, blah. Resource poor setting is not that, but that is a resource poor setting. But resource poor setting is a farm in Australia, where it could be 2,000 kilometers away from the next town that contains a good lab for diagnostics. So resource poor settings is not about being in a rich or a poor country. It's being in a place where you do not have access to technology. That is what it is. So what do you need in those resource poor settings? The first thing is you need things that are simple. Any kind of complications, it will essentially nullify any kind of effort you want to do. Second, you don't have molecular biologists everywhere. So you should be able to do it with personnel that is not farmers. That's impossible. You can't ask farmers to do these diagnostics, but you don't need to have somebody with a degree in molecular biology, somebody who knows, but doesn't need to be specialized. At the beginning, we started doing, we want to be able to do this in the field. And then after years, we've been working on this for more than 20 years now. We realized that it's a lot better to go to the field, get the samples, and go back to your hotel. And in the hotel room, have a nice lemonade and have air conditioning, and then do the testing. At the beginning, we were always obsessed doing it in the field, in the back of a car and like that. And then we learned, what's the point? You know, when you are in the field, you essentially are sleeping in a motel or something, so you can always get it back. You need to be practical, all right? Your equipment needs to be portable, and portable means portable. There are some equipment that they tell you is portable, but okay, let me explain this. A piano is portable. Means, yeah, you can take a piano to different places, but it's not comfortable to move a piano. So by portable means something that you can really put perhaps in your pocket. You need to be able to get reagents that are easy to store, if possible at room temperature. And then you need to have some kind of kit that you can distribute. Once you develop new technologies, you can distribute that to your collaborators. So the methodologies that the main methodologies in general terms, one is the classic one, classic pathogen identification, where you look at the disease symptoms, you know, you isolate the pathogen, those are the Koch postulates, you know, but for that you need a good plant pathologist, and it's quite slow. Now, I don't know in Indonesia, but in Australia, we are running out of classic plant pathology. As a matter of fact, we are actually hiring pathologists from overseas because we just don't have enough in the entire country, because young people don't find that a sexy kind of occupation. They all want to be lawyers or molecular biologists. So a classic pathologist that look at a plant and say, oh, that looks like a viral disease, it's difficult to find. Of course, then you go into more kind of technologically advanced method, like antibody-based, and you have the ELISAs, the lateral flow strips, and like that. What are quite okay, but there is one problem, is that you need a constant supply of antibodies. Now, constant supply of antibodies sounds easy to find, but it's not, especially when you need a lot of it. For COVID, yes, you have as many antibodies as you want. For the sugarcane strike mosaic virus, you know, a commercial company will look at you and say, I'm not wasting my time with that. So it's not that easy. And then, of course, you have the molecular assays. Those molecular assays are mostly amplification-based, all right? They're very sensitive, very specific, and very fast. But you need really good experts, and you need specialized equipment. And that specialized equipment is expensive, and it's not very portable. All right, so when we talk about, of course, in my lab is a molecular biology lab, so we are mostly concerned about, you know, nucleic acid-based diagnostics. So when you think about those kind of diagnostics, there are three steps. There are three steps of that. One is sampling. You need to go to the field, and you need to get your sample, your tissue, isolate nucleic acids in order to amplify. Many times you need to quantify, and of course you need to get someone who knows how to isolate DNA or RNA. DNA and RNA are not easy to isolate. Then you need to amplify, meaning you need your machine, you need a reliable power supply, and when you work like I do in places like PNG or Lao, a reliable power supply is not guaranteed, believe me, all right? Anytime the power goes off, if the power goes off, the PCR says, boom, that's it, I abort the ground. And of course, you need a lamp. And finally, you need to know whether it worked. So you need to look at your results and say, well, did it work, yes or no? Most of the time, what people do, and this is not qPCR, because qPCR is way too much to do it anywhere in the field, all right? So you need to do an electrophoresis and then use UV to, you know, look at your DNA amplification. So I will tell you our first effort. Our first effort, and this is, we're not using this anymore, but it's one that we have published, and it was how it all started. We decided, okay, let's divide 1, 2, 3, the 3 steps. For sampling, we started using SPRI, and the SPRI are the typical magnetic beads that can bind DNA and RNA. And essentially, you only need a magnet in order to do your washes and whatever. So we use that. Then, for amplification, we decided to run away from PCR and we went into isothermal. Isothermal, in my opinion, is almost essential if you want to do anything in the field. Because anytime that you need to get a PCR machine, even though they said there are portable ones, they use a lot of power, it's very difficult to use them. In our case, we started working with RPA, recombinase polymerase amplification. This is relatively recent isothermal technique, means you don't have any cycles, and you do it at 37 degrees. 37 degrees, that actually means that it's relatively easy to get that temperature. As a matter of fact, I got some of my students and I actually taped tubes to their arms and I asked them to keep it in their arm for half an hour and then we analyzed the results. It's not as good as a water bath, but you can make it work by just having it in your body because the human body is around 37 degrees unless you have COVID and it's 41. All right, okay, and it's quite fast. Actually, and the time of amplification with this, anything between 20 to 30 minutes, you get amplification of your product. And then we came up with, how do we visualize our results? And in that respect, we actually started inventing things. In this case, we invented something that is called bridging flocculation assay. And that is that we managed to modify the paramagnetic particles in a way that when there was amplification, the particles would drop to the bottom of the tube. While if there was no amplification, and you can see here what it says healthy versus sick. Healthy means no amplification. If there was no amplicon of DNA, the paramagnetic particles remains suspended in the tube, while if there is amplification of DNA, they all drop to the bottom of the tube. Now, by doing that, then we started looking at, obviously, we started with plants, all right? So we started with Pseudomonas, and this is, you can see, this is Arabidopsis thaliana, this is Pseudomonas syringae. In the lab, you see at the top, you do your, this is a normal PCR plus your electrophoresis, and you can see that you have your bands there. That's okay because they're a lab. Then on the SDG, the single drop genomics, what we did was RPA at 37 degrees, isothermal, and then flocculation. And you can see that whenever you have a band in your gel electrophoresis, you will have the drop of the paramagnetic bits to the bottom of the tube. So that was one good visual way of having a look at it. I know that there are people using a lot of pH-based, pink versus purple and like that. Let me tell you one thing. When you are in the field, pink versus purple is impossible to tell by the human eye because you're not in a perfect lab with white backgrounds, etc., etc. But this was quite good. Then, of course, we went to Fusarium. We work a lot with Fusarium. It's a disease that is very common for a lot of plants. You can see at the top, you have your laboratory results where you can see that when you inoculate plant with Fusarium, after one day, you don't see anything because Fusarium comes from the root. So it needs to move up the plant. But as soon as it gets to the leaf, you can see a band in your laboratory with an electrophoresis. And with the SDG technique, you can see that there was dropping of the paramagnetic particles to the bottom of the tube. Botrytis is another very common disease. Also, we did try the diseases that we already had in the lab. That was good for us. And then we went for RNA viruses because unlike humans, plants have a lot of RNA viruses, more than DNA viruses. So we did it, and then you can see in the lab that when you have reverse transcription on the left-hand side and a positive, you can see a band. And of course, then you can see also the dropping of paramagnetic particles in the tube. Then we were about to publish that, and I remember that I woke up in the middle of the night and said, this is a platform technology. It doesn't care if it's a plant or if it's an animal. So we actually found some friend of mine, and I asked them for any animal disease. And they gave me this one, bovine respiratory disease. And I don't even know what it is, but they gave me some samples and we analyzed it with two different set of primers. And as you can see with the TK1, this herpes one, you can detect it with normal laboratory techniques, but also with our newly developed techniques. And when you detect a different protein, glycoprotein B, is the same thing. Again, we were about to publish it, and I thought, like, hold on a second, can we give it a bit more relevance? And I actually called a friend of mine that works at the hospital, and I asked him, can you give me some HIV? And of course, he said no. I cannot give you any HIV, all right? So I did send my student to his lab to work with HIV positive samples in his lab. Obviously, it's high secure lab because it's a important disease. And as you can see, we were able to detect HIV with our technology. Once we started doing that, I said, oh, this is it, Once we started with them, we went to malaria. So we got, in a different lab, we got some samples of malaria, and we could see that we could also detect it. Finally, okay, here it is, tuberculosis. Again, we were able to detect it. Why did we do that? Because our lab works in platform technologies. We are interested in plants. I normally always make a joke that we don't like humans, so we only work with plants, all right? But the truth is, when you develop platform technologies, it can be applied to pretty much anything, because DNA is DNA. And nobody cares if it comes from a cow or from a tomato. So that's what we did. This is influenza that was before COVID. But now it has a special meaning here because you can have influenza, and then we even tested E. coli contaminated environmental water, and we were able also to detect it. Meaning that the whole thing had a multiple dimension, all right, and it all came by combining two technologies that were already invented and a third one that we had to develop ourselves in order to visualize it. We publish everything. You can see this in Chemical Communications, and they gave us the cover of the journal. By the way, this eye is the wife of the postdoc who did most of the work, but he changed the color because she's got brown eyes. But her husband thought that having green eyes would be better for the cover of the journal. She wasn't happy about it, but it was good. So we were really happy. But the truth is, we got this paper is cited by a lot of people. We made a good impact on it. We got the cover. But the truth is that I was not being honest with people. We were claiming that we're going to revolutionize this and that, like you always do. Good scientists always exaggerate. The meaning of their findings. But SDG had a number of problems. The first of all, if you use paramagnetic particles when you're in the field, you still need micropipettes. You need to pipette a lot. You can, micropipettators for people who work in molecular biology is what we do every day. People who doesn't work in molecular biology don't know how to use them, or they use them improperly, okay? The other thing is, there was a lot of manipulation to be done. Even though I did send my student to the garden, she did a lot of assays sitting in the grass in the garden at UQ. But then she was coming back to me and saying, Jimmy, it's a lot more difficult when you're sitting in the grass. All right. Okay. Then the amplification, funny enough, and people doesn't know it, but we already published a paper on this because I thought people should know about this. RPA will always amplify something. It's only a question of time. Even in a reaction without any sample, you will get amplification with RPA. And the funny thing is that the discoverers of RPA actually mentioned that in a really small phrase in their first paper, but nobody remembers that. So The problem is you will amplify okay in 10 minutes, but if you wait 20 minutes, you will always get amplification. So the problem becomes, is what I got, when do I stop a reaction? Is what I saw, if I have very little pathogen and it comes at 18 minutes, is that pathogen or is it self-amplification? So RPA was a bit of a problem. And because it's only sold by one single company, it's expensive. It's an expensive exercise. And the visualization of results, it will always work. But the truth is, apart from my student and the postdoc, I gave it to other people in the lab, and they were not able to get the things working as well. These two people were able to do it. They were not lying, but they had done it hundreds of times, meaning you needed a lot of experience. So we said, okay, we need new systems, all right? And we started by inventing something, is what we call the DNA dipstick. You see that in the figure here? I have a handle and what we call the nucleic acid binding. So this dipstick can be used in this way. Let's say that you have a tissue. At the beginning, we started using ball bearings just to break the tissue. Anything that can break the tissue. So all you need to do, by the way, this, what it does is purify DNA in order to load an amplification reaction. So you come, step number one, you dip your dipstick into your sample. Step number two, you wash it in a wash buffer. In this way, the DNA stays in the dipstick, but the contaminants come out. And finally, you dip it into your amplification reaction. That is the entire purification system. Now, I have a video because I got my student to do it. Say, you need to demonstrate it. This is a video demonstrating how is used. And by the way, I asked them to have a laboratory clock in here. You have your plant sample here, the wash buffer, and the PCR tube. So you need to disrupt your sample. If you got ball bearings, you do it with ball bearings. Anyway, any way is good. And this is the purification. One, you put in your sample. Two, you wash it. And three, you put in your PCR reaction. And that's it. You can see here that the entire purification system is 10 seconds on the clock. And if you compare that with kits and expensive kits and like that, you say that this is one big thing, all right? We tried a bunch of different tissues. We started with typical rice, tomato, sugarcane, sorghum, soybean, barley, wheat. We were able to obtain amplification. This is PCR. Then a friend of mine told me, those are easy ones. You need to go for fruit trees because leaves from fruit trees have a lot more secondary metabolite. So we tried mandarin, lime, lemon, and passion fruit. And the reason is because those were all the trees that people in my lab have in their gardens. We didn't pick them, we just went for whatever we could find. And then we said, as we did with the previous one, we need to look for more than this. So we went for human blood. Human blood is notorious for having too many inhibitors of amplification. And you can see here that without purification, if you put 1 microliter of human blood into your PCR reaction, you don't amplify anything. But when you use the dipsticks, you get amplification. All right? And this is a melanoma cell line, again, that was asking favors to people around the university. Give me something else. I want to try more things. And as you can see, it works quite well. The fact that it works in human blood has a special relevance because this means that you can load a PCR reaction for pretty much anything you want that is measured in the human blood. You can load it in 30 seconds. Of course, we went back and then we went into pathogens because the first results were just, genomic. This is pseudomonas aeruginosa, and you can see here that you are able to amplify it. These are PCRs. Then again, we went asking people, give us disease, something. They gave us some disease from pigs, lung swabs from pigs and saliva, essentially. If you don't purify the saliva, you cannot amplify anything. In this case, it's Actinobacillus pleuropneumonia. I don't even know what that is. Okay, it's just something we knew the primers, but if you purify with the deep sticks, you can see that you have a super duper amplification product. And even better, we tried what happened if we try with RNA. Can we purify the RNA? Yes, we can. This is in tomato. Trying to pronounce, yep, in tomato, CMV. This is infected plants versus healthy plants, and you can see with reverse transcription, you get a positive in the infected ones. Without reverse transcription, you don't get anything because it's RNA. And then LAMP, isothermal amplification, loop amplification. You can see that you get amplification in the infected tissue, but not in the healthy one. That actually means you can detect RNA virus. All right. Then what we did is let's compare it with commercial stuff. And we got the paramagnetic beads. That was the ones that we used in our original method. To compare them. With the deep sticks, it takes around 30 seconds to do the entire purification. Paramagnetic beads, it takes around 15 minutes to do it. The pipetting steps with the deep sticks, and using the method is 0. You don't need to pipette anything. With the paramagnetic beads, you need 6 pipetting steps. And the cost is around 15 cents. For the deep sticks is 65 cents for the paramagnetic beads. And 15 cents includes the cost of the tubes. The actual deep sticks is a lot better than that. Then we also compare them. So then you have the deep sticks versus paramagnetic beads. And what you can see here is this is purified DNA. And you can see that with the deep sticks, you can get 1 and 0.1. That is essentially the same sensitivity that paramagnetic beads. These are dilution series. And then from crude samples, these are not purified, these are crude samples. And these are again dilution series. And you can see with the deep sticks, you get up to 0.5 with good detection. While paramagnetic beads, when you get to 0.5, the detections are going down. Now, let me remind you the cost of a bottle of 450 mils of paramagnetic bead costs $6,600. Okay. We published that. We published in PLOS Biology, not PLOS, but PLOS Biology is a really big impact index, and I want you to, it's available for all of you. I want you to have a look at the number of views that we've got so far. Over 150,000 views of people doing that. Now, this method is now being used by, I don't know the number of labs using the method. No, but not only that, there are companies, biotech companies, using our deep sticks for their work. All right, that was patented at one time, but then we let the patent expire, so it is not patented anymore. And I'll give you some examples of things that we're doing with these deep sticks. This is coconut. This is sawdust. There's a phytoplasma disease in coconuts, and we use this with our deep sticks, and it works quite well. This is cassava. And in cassava, as you can see, first, you need to optimize it a little bit, like, how much tissue, how much buffer you put, like, 1 gram versus 2 mils of buffer, or 1 gram with 5 mils of buffer. So there is always optimization to be done. This is, in our case, we use leaf discs because we can cut the disc with the tube. So you can put a cassava leaf on top of the tube, and then with the leaf, you cut the disc, and it's very easy, so you don't need to have new punches or anything. And then we optimize our shaking time. You know, what about 5 seconds, 15 seconds is all right. 15 seconds is more than enough to see that you can get amplification. But then, all that is good, but how do you know if it works? You need to run the reaction and you need to visualize it. So for that, what we did is we pretty much invented a machine that we made it in the lab, all right? This is not for PCR, it's for lab. Meaning it's amplification, isothermal amplification assisting fire studies, all right? This is one of our previous models, not the one that we have now. We call it the Diagnostic droid. Okay, and this is a GP and a student who was developing all this. At the beginning, I will stop this here. As you can see, this looks like a coffee mug because it is a coffee mug. For this first iteration of the machine, we bought it on the internet. These are the coffee mugs that you plug into your car to keep your coffee hot while you're driving. It's exactly the same, but we modify it inside, okay? And in this iteration, we made one very simple, only for detection, and then they communicate with your phone. You can see here the insides of it, you know, it's got LEDs, detectors, and everything. We did an app on the phone that was able to communicate with the device. And the device start heating up, and if you look at the temperature downstairs, 62.9. When it gets to the 63 to 65, you just load your samples and press start on your phone, and the machine will start just testing the samples and checking whether there is amplification or not. And then the best part is that you don't need to know it. The machine will tell you this is positive or this is negative, right? That was our version 1.0. What we have now is very, very, very different. This is what we have now. It's still in a coffee mug. It's still in a coffee mug because we found that it's very easy. This one, you can plug it to the car lighter. So you can actually take samples in one field and then put it on your car and drive to the next field. And the machine is analyzing the samples in the meantime. But this one got an LCD screen. Instead of 6 now, it analyzes 8 samples. And it doesn't need a phone app. It's autonomous, all right? I will interpret your results, and it got a memory card that actually gets all the data during the entire lab. And then you can put it on your laptop and analyze your data. It actually measures more than you need. This is my example, and this is in PNG, coconut lizard yellowwing. The truth is, I was going into the jungle, you know, cutting some palms and drilling holes in the palms and getting some sawdust. And then, you know, getting the machetes back and going back to the back of the car. And this was my lab. This was my lab. You can see my tubes in here. And this was the first version of the diagnostic droid, you know, plugged into the car. We have done quite a lot of work on that, and it's quite reliable, all right? But then we thought, what about if you have no power? All right, so what we did, and this was in Cambodia, is just get a big styrofoam box. You don't need the fish. You can eat the fish for dinner, and then you keep the box. All right, and we have personally heated water on the fire and then mix hot and cold water. When you put that on a styrofoam box, it will keep a 65 for more than an hour. It just keeps, you know, heat very well. And then you float your tubes. But the problem is, well, this is not the machine. So how do you know if it works? So we invented another thing that this one is. I love this one. This is nanoparticle-based flocculation that has got nothing to do with the first one. This is like 100 times cheaper, faster, and better. I want you to look at the tubes. When you finish your reaction, you add our nanoparticle into each tube, and it's black. And anything that comes down means your tube is positive. If it doesn't come down, your tube is negative. I'm gonna, because it's only 10 seconds. I want you to have a look. I'm gonna run this again. So I want you to have a look at the tubes and see how things are going down the tubes. You can look at these two tubes and see how the particles are coming down. By the way, if you wait 2 hours, even the negative will end up coming down because, nanocarbon also waits, okay? But if you shake it again, it happens exactly the same. And if you wait 3 days, it happens exactly the same. It's in solution or it goes to the bottom. About refrigeration, we actually have developed methods to lyophilize our reactions. We can stabilize them, we can keep them with us at temperature that go all the way in my luggage. Brisbane, Bangkok, Vientiane, they still work, all right? So work for more than 2 weeks. This is one of the examples. We did E. coli contamination in fresh vegetables in Laos and Cambodia. We brought everything in our luggage, and this is me getting extremely strange vegetables that I have never seen in my life. We went to a lot of different markets, got vegetables, and then we analyzed them. And these are the positive for E. coli versus the negative for E. coli. It's funny what can you learn from this? We actually learned that 50% of all vegetable samples in countryside markets was contaminated with E. coli. But in the cities, like Phnom Penh, only 5% were contaminated. So people in the countryside were getting a lot more disease because of the conditions of the markets. It's nothing to do with the vegetables, it's got to do with the markets. It's funny because that's what I thought, that they're immune. They're not that immune. They still get sick, but they don't report it. They get sick so often that they don't report it. So what have we done with our technology? For example, the poultry industry, we finished this project for Campylobacter bacteria in chicken carcasses in factories when they prepare chicken. They're using it in PLG. We've done it with Coppola syndrome. Mexico and the Caribbean, they have actually organized regional workshops to show how the deep sticks work for other people with other crops, all right? In Cambodia and Vietnam, we're working on cassava, and this is ACR funded. I know that in Harvard, they've been using it for human viruses. People at the hospital in Brisbane have used it in remote islands in the Philippines to test stomach parasites in humans. Because it looks like in the Philippines, some islands they need to get on a boat to find a doctor, so it's easier for them to go. And now, just as a goodbye, let me introduce you to my university. You guys are more than welcome to come anytime you want. I'll make you feel welcome when you come to Australia. This is UQ. I know Australia, I'm Spanish, but I've been away from Spain for most of my life. The day after I finished my PhD, I left the country, never went back. At UQ, I've been since 1995. It's actually a really good university. Take into account that Brisbane is a very small town. It's not a big town. It's around 2 million, I think, or something like that. But if you look at it, our university is in the top 50s on pretty much any ranking in the world. So it's a good accomplishment. This is one of our campuses. It's a beautiful campus. We got 3 of them. Around 55,000 students. 20,000 of them are international. Around 20,000 postgrad students. In agriculture, we're #1 in Australia. We're number 26 in the world in the QS rankings, but we're number 3 in the world in the scientific publication rankings. We're number 3 because number 1 and 2 are always Davis and David. All right, that's we will never ever do better than them. Okay, but it's not a bad thing. This is my state. It's Queensland. It's a very tropical state. We have beautiful beaches. We have a lot of islands. This is just to give you an idea of my state, that is not even the biggest one in Australia. We got almost 2,000 islands, 1,700 beaches. But let me explain this. In 1.9 million square kilometers, that is the size of Indonesia, we have 5 million people. Yeah. Okay, that you imagine Indonesia with only 5 million people living. I just looked it up yesterday. Jakarta has 11 million people
during the day.
That is Jakarta. Okay, so it gives you an idea that Queensland is so low populated. We have an apartment at the beach, and it's south of the Gold Coast. Gold Coast is for tourists, so that's very populated. But in my beach, it's around 20 kilometers, and when we see more than 5 or 6 people in 20 kilometers, we go, "Oof, too many people." We're going home. And then it's got, if you like the water, and I love the water, I'm a sailor, and I work very hard, but if it's windy, I get my kites, that you can see here, my kite, and I get my boards, and I go to the beach. In summer, essentially, I go to the beach every day. I go to work at 5 in the morning, but by 3 o'clock, I stop work, I head to the beach, and I spend 3 or 4 hours sailing every single day. And this is such a high quality of life, all right? But by the way, for the young people that Bueni was talking before, this is not for you. This is only for me. My students are always working. And of course, pretty much we have a lot of collaborators in very different places. We do work a lot with the Chinese Academy of Sciences because of my attachment as the professorship that I have over there. But we do work a lot with Singapore, the US, Shanghai, Malaysia, and hopefully will be Indonesia the next one in this list. So with that, I thank you very much for your attention. I'll be happy to answer questions.
Okay. To see my face here. How to maximize?
And here, please, you should carry here.
Yeah. Okay. Yeah. Thank you, Jimmy. It's very, very interesting talk. I'm sure you all very wide awake now, wide awake to listen his talk. And we are here hybrid in the seminar room at the building of the this organization for life sciences and environment. So if you see Professor Jimmy Botella's face coming to this way, to the other way, and zoom and your camera, because he's talking to us here on site. Yeah, so yeah, I just want to summarize a little bit that no matter how advanced your science or technology or expertise, you still need to think something that workable or implementable on the ground, that people or users can use your technology, also be beneficial to other people. But still, we are still optimizing our work, our technology in the lab. So here, I will all learn about another abbreviation of SDG, because we know the SDG is the Sustainable Development Goals, and in Bahasa Indonesia, in Indonesian language, we we have abbreviation of SDG, short form of sumber daya genetic. Sumber daya genetic means genetic resources. So we have all different kind of SDGs abbreviations. So here also that we learned that he and the group in Queensland simplified many methods. It's reduced the cost, it's reduced the time, and no need liquid nitrogen, no need kit that is very expensive. We are practicing now, Burati, to report to you that in now it's 5 days already. We already did, we've done several things that we trying to simplify things. So hopefully it will be our property right patent soon. Yeah, because we also combine many things, many procedures becoming only one-time procedure. And also here, what he has explained to us or shared with us is that the technology is not just worked only for plant, but also for animal, also for human, and for human also all different kind of diseases. So all of us here at genetic engineering and others who actually attend this seminar so can apply and ask him later on how it worked, because it's very useful for us to know. And also that comparing in plants, working with annual crop and also perennial crops, yeah, it's different. It's much more difficult to work with perennial crops because it's got the polysaccharide and also polyphenol and metabolite, secondary metabolite contaminant. Yeah, so the invention is very important. Yeah, so keep doing new innovation. Of course, innovation is new and new things. Yeah, that now that we don't need to keep it in at minus 20 or in the refrigerator, but it can be in just room temperature. So I think this one is very interesting. So now it's all open for discussions. So I don't know, is anyone in the chat? Yeah, I'm going to open it. Okay. So there are 4. One person asking for 4, or it's already, or it's just 1. Okay, 4. One, this is question for Prof. Jimmy Botella. One is from Nurfianto. Do samples to be tested need to be pre-treated first? Okay, number 1. And second, how is the sample preparations and use of SDG for plant diseases that have several pathotypes, such as rice bacterial leaf blight or Xanthomonas oryzae? And then number 3, he got 4 questions. How is SDG applied for plant breeding purposes, especially for biotic stress tolerance due to pests and diseases and abiotic stress? And number 4, I don't know whether you can remember, but I can go back here again later on. How is the application of SDG for aflatoxin and mycotoxin contamination in agricultural products and its prospects for use by the quarantine agency? Thank you. I think this must be from the Ministry of Agriculture. I mean, used to be working at Ministry of Agriculture. So I'll let you answer.
That is not from number 4.
Okay, number 4.
Look, the interesting technology is useful to purify DNA from the original state from a sample to amplification. Therefore, it's not good for chemical groups. If you want to do aflatoxin, this is only good for anything that you can amplify, meaning nucleic acids. Therefore, I'm afraid that toxins like aflatoxins or mycotoxins are not amenable to this, because this is always linked with amplification. Therefore, you can only do anything that needs amplification. how is SCG applied for plant breeding purposes? Well, plant breeding purposes, it can be extremely used, and there is a lot of breeders using this process now. But the way you do it is if you have a QTL that you know, and you're following anything that you can amplify, you're looking for a gene and you're following the gene as a QTL for any kind of yield or whatever it is, or biotic stress or whatever. Again, you can use this because essentially it's so fast that you don't need to do, sit up extractions or anything. So it will allow you to do lots and lots of analysis of your breeding population, but you need to know what gene are you looking for because you need to amplify it. Number 2 is sample preparation for different pathotypes. Well, you can do any pathotype you want, essentially. All you need to have is the power to amplify it, meaning you need to know the primers that you are going to use to amplify one specific pathotype. What this method will do for you is make it extremely simple. The amplification, you will need to find out, to optimize your primers and your amplification conditions. But what the method would allow you is to do it, pretty much anywhere you want and for next to zero cost. Yes, samples do not need to be pre-treated. As a matter of fact, you thought that we were checking it in a tube, but we don't do that anymore. And now what we do in cassava, for example, is we cut a little bit and we put it on a plastic bag, put a bit of a destruction buffer, and then use a pen and just smash it inside the plastic bag. And then we use the dip stick, we dip it inside the liquid in the plastic bag. So not even a tube anymore, but a plastic bag, anything that you smash. Sometimes I do have witnesses here who have done the whole process and have been able to amplify it. And you did it with your fingers, no? In a plastic.
With a nail. It's the third.
Yeah. In our case now, as part of the simplification, we use a little plastic bag around, 5 centimeters by 7 centimeters. We put 1 or 2 mils of buffer, and it doesn't matter if it's 1 or if it's 2, it doesn't really matter. And then we put the tissue inside, and all you want is to break the tissue. So you don't need to protect the tissue first.
Okay, I hope that answered your questions. Otherwise, we can go back later when we have time. Although we still have time for like 40 minutes, right? To 3 o'clock. This for new messages.
Sometimes it's good, yeah.
Okay, this is Desviani, Univ. Yeah, thank you for his comment actually. It is very inspirative and very interesting presentation. My name is Desriani from Research Center for Genetic Engineering. We met yesterday at the PCR room, last time. She has questions. Also, how many? That's Number 1, please correct me if I'm wrong. From your slide or video, you explain about dipstick for avoiding from contamination problem in the field. Is it correct? How you develop this dipstick? It must be a long story. Is it the same thing as lateral flow strips? Yeah, just one by one first.
No, it is not the same. I might have because I can show. I brought some with me. Can you see it?
This camera.
Yeah, this is stage, all right? So what you do with this, it's not a lateral flow device. You put it inside your tube or your bag, anywhere that you have a DNA solution, and that will bind the DNA to the end of the dipstick. And then this dipstick will have DNA, but it will have contaminants too. Therefore, what you need to do then is put it on a second tube where it's going to wash the contaminants out. And then finally, you put it in your third tube, that is the PCR tube or the lamp tube, that it will release the DNA into the amplification tube. And then you run the reaction.
Okay, and next.
Two fingers.
Yeah. Okay. What kind of software do you use in order mobile phone could read the result of DNA amplification result?
Yeah, we did develop our own software for that, but it's not needed anymore. It's not needed anymore because we don't use a phone anymore. Our device now is self-sufficient, so you don't need any communication. It will send you messages with everything that it needs, and it will communicate with you without an app. I mean, making an app for the phone is not easy, but it's not that complicated.
Okay, with this, I hope that answers your questions. Also, he still will be here until next week, so you can come and discuss with him directly. This is from Afif in Malang. This is also from our group. Yeah. So thank you, Prof. Jimmy, for the very interesting message for us. One thing that I would like to ask is, do we need specific primers in the screening for the disease screening? Thank you. She must have missed. Yeah.
Yes. Right. Yes. The primers again. Whichever primers, depending on the pathogen, you will have primers for it. Most of the pathogens already have primers developed by other groups. So for Fusarium, we didn't invent the primers for Fusarium. We just went to the literature and just copied two primers of somebody who has done it. For Pseudomonas, we didn't develop the primers ourselves. We just went to the literature. What our method allows you is to get whichever primer somebody has developed, and then yes, do it in a very fast and easy way. You know, get all the way to amplification in a very fast and easy way. Now, if you need to develop primers for things like LAMP is a very difficult technique to develop primers. LAMP uses 6 different primers. It's not like PCR. PCR uses 2 primers, LAMP uses 6 primers, but there is software that will help you develop primers for that. But my advice, like I tell everyone, don't invent anything if you don't need to. If somebody already have primers to amplify Fusarium, use the primers that somebody has already developed.
Okay, Mbak Afif, you should come here. Sorry, so from this side it's a bit difficult.
Sorry. Yeah.
Yeah. Okay. Oh, this is for the absence. Oh, going down, maybe there's another one. Oh, there's Riani. Oh, only just thank you for the explanation. Thank you very much, Prof. Annie. Yeah. Any other questions? Yeah, as Professor Jimmy already explains to us, that is very useful for also plant breeding purposes, including us at the plant genetic engineering and genetic engineering research center, because then it saves the time at the beginning, yeah, for extraction the DNA and then or RNA, yeah, so it saves the time. And we don't need the expensive kit. Wait for our result and publication soon. Yeah. Okay. Any other questions? We still have plenty of time. Maybe from here on site. Yes. Yes.
I would like to ask questions. For the
Nama perkenalkan.
Dilan.
Yeah.
For the DNA isolated by the dipstick method, how do we know if the DNA is in good condition?
Okay, good, very good question. So the question for those who might not have heard it, just in case, was how do we know about the quality of the DNA? You will know during the optimization process, when you start with a new plant, for example, cassava, all right? So what we do is we use the deep stills and we amplify genomic DNA. And because if you are looking for a pathogen, you never know, is the plant sick? Maybe it's not so sick. So during optimization, what we normally do is we test genomic DNA from the plant, for example, from cassava. Once that we know, if you put like a 1 by 1 centimeter piece of leaf in around 2 mils of extraction buffer, all right? And we're able to amplify the cassava DNA, then if you have a pathogen, you should be able, because that actually means that is like your positive control. Positive control means you're able to amplify the genomic that will always be there, then you're okay. Now, the pathogen is just a different kind of DNA that you will have there. But again, when you have a sick plant, you might be very sick and have a lot, or very little sick and not have too much. So we always optimize it with the genomic of the plant that we are trying to analyze. And after that, we never change the conditions. By the way, you purify DNA, but this is not like a kit. You will not be able to get that pure DNA into a tube and take it. You can only get it from your plant to the amplification tube. Okay, it's important that you understand. By the way, because it takes 30 seconds, you can get the same extract tomorrow and do it again in another 30 seconds. You don't care, you know. If you use a kit and it takes 3 hours, then tomorrow you don't want to purify another 3 hours. But with this, you can do it as many times as you want because it takes almost 0 time. Meaning that this will not give you 500 micrograms of DNA in a tube. It will lock your amplification reaction. And before anyone asks, the diagnostic droid is homemade, made in our house. I wish I had to share, but we don't. What we are doing now is I have hired an electronic engineer that is redesigning the machine in a way that we can send it to a factory to manufacture 100 of them. What we didn't realize when we made this in the first place is that one thing is making something in your lab or in your house, and a different thing is sending it to a factory. A factory requires all the drawings in a specific format, and they cannot do like we're soldering more than 50 different connections there. A factory cannot do that. I didn't know, but I've learned now from the electronic engineer. So they needed to redesign the entire machine, because for factories, robots are the ones that will be doing all that. So hopefully, we're working, with ACR and Jono. If we get the redesigned manufacture diagnostic droid, we might try. Jono is trying to get money from the Asian Bank. It's called that Asian Bank Development Bank. ADB. Yeah. Because if we're able to finalize that, we will try to get money from the ADB to make a run of 100 of them, so we can start sharing it with collaborators.
Okay, I think there's another question here. This one is, Nurmala. There's a question from Nurmala. Is lateral flow assay has to be made from monoclonal antibody? If the antibody for lateral flow from recombinant polyclonal antibody, is it sensitive enough to detect samples?
Okay, well, it's got nothing to do with what I say, but I know about it. Lateral flow devices, you can make it with monoclonal or polyclonal antibodies. Which one is more sensitive? It depends who you ask. Some people will tell you the monoclonals are more sensitive, some people will tell you the polyclonal are more sensitive. One thing I can tell you, the polyclonal antibodies have more epitopes because they are made to more parts of the protein. Therefore, you should have more antibodies bound to a single molecule of protein than the monoclonal antibodies. On the other hand, the monoclonal antibodies, you will always have the same title. While in polyclonal antibodies, you might get a batch that is super good and a batch that is super bad, and it might not work at all. Lateral flow devices are great, but I tell you one thing, the Australian horticulture glasshouse industry came to talk to me because for 6 months, all the lateral flow devices with the antibodies that were buying from the UK came defective. So that batch of antibodies was not very good, so they couldn't detect anything. And that is the problem with antibodies. Antibodies is very difficult to standardize the quality that you have. Of course, if it's for COVID, where pretty much there is no barrier about how much it costs, you will have it. But for, as I said, ratoon stunting disease in sugarcane doesn't produce enough money for a super big factory to give us, a constant supply of antibodies.
Okay, that's it. So any other questions here on site or online? Ada yang mau nanya lagi? No? Okay. I think yeah, this one for my research group, actually this is the second technology that we got from our collaborator that can be applied in the field. So it's simplified things that we don't need to bring the samples to the lab, especially if the samples from the conservation area in Indonesia. From the conservation areas in Indonesia, we need a special permit to enter the area and also to take out anything from the conservation area. Okay. And they count one sample each, how much, yeah. And also need special permit, although the permit last time from our institute, Indonesian Institute of Sciences, and now BRIN, we are the scientific authority for giving for biodiversity. So this will simplify our life, make our life easier, because we don't need to take the sample out of the area. Yeah, so one is the technology from Rutgers University from the US. So we could analyze antioxidant or flavonoid or other metabolite from the part of the plants in the field directly. Yeah, whether it's can be for antioxidant, can be fungal or bacteria. So that's, and then now we already all learned from you that this one also very practical if we want to do it in the field. Yeah, so that's another question. So we can go back to that again. Yeah, Bu Weni. Thank you, Professor Jim Botella. Perhaps you have any experience in DNA isolation of woody plants. May you can share your tips for getting good DNA from woody plants for sequencing. Yeah, I mentioned already the perennial plants is actually the like woody plants, yeah, perennial or trees, yeah, or forest trees. It's more difficult to get nice quality, good quality of DNA because of the polyphenol, the polysaccharides, or other things. So she asked whether you have tip.
my first trip is don't work with those plants. Yes, try to work with tomatoes. But if you don't have any other option, look, the problem with woody plants is always going to be polysaccharides and phenolics. Polysaccharides and phenolics, there are a bunch of different, purification methods that have been developed for it. We have used one that we developed for pineapples for a long time. It's worked very well with woody plants. Again, woody plants, I'm talking about woody plants, I guess you're talking about leaves, okay? Because you don't want to get it, into the stem or anything. And these deep sticks will work with woody plants, I've been able to amplify things from sodas. Okay, that is in coconut trees, drill a hole and get the sodas, put some buffer into it, and then use the deep stick to amplify. But this is to amplify, it's not for sequencing. If you want to do sequencing, and I'm talking genome sequencing or whatever, then you need to go through one of the complicated methods that are in there.
Yeah.
Now, if all you want to do is PCR and then sequencing your PCR because you want to look for genetic diversity or things like that, then this method is perfect. You can use the deep sticks to do your PCR, and then yes, send your PCR for sequencing because we've done it 1,000,000 times and it works perfect.
So yeah, it's depend on your purpose. Certain purpose you can just use dipstick, and then Professor Botella mentioned that it worked well also with other species. So yeah, last time because I've been working with woody species also since I got back from UK last time. So yeah, we need to do more effort than working with agricultural crops because it's well simpler because I work with both type of plants, so I can compare. But yeah, sometimes we also put something like PVP in the extraction buffer so that it can reduce the polysaccharide and phenolic compounds. Okay, learning. Yeah, any other questions?
Oh, by the way, if anyone wants to produce your own deep sticks, right? The old way, these are the plastic ones, but the old way, kind of desperate to do it yourself, is published in Nature Protocols. So we have a paper in Nature Protocols explaining this is the old ones where we used to use wax and a spaghetti mirror. So it's not very professional, but it works well, right? So you use wax and a spaghetti maker to do your deep sticks. These plastic ones are a bit different, but it's the same principle.
So that means we can produce.
You can produce.
Yeah. Okay, that's the way how to have new things as an innovation. Okay, if there's no other questions here, on site here, no. And so thank you very much. Last offer, last offer. Last offer before I'm going to give to hand over to the master of ceremony. So if no, then thank you very much for all your attentions. But maybe you have a closing statement.
Oh, well,
Yeah.
I will leave a bunch of activities here. If anyone wants to have a personal look at them, Professor Eni, I will leave 30, 40, or 50 of them with her. In case that you want to have a look, they're not going to be clean, so I wouldn't use it for any experiments, but at least you can have a look, so Professor Eni can provide them to you.
Okay. Okay. When you come to me, I will get the fee to see the. Yeah, I'm just joking. Yeah. So we have to be balanced with him because he's Spanish. So this is the way the Spanish man talking. So yeah, I'm already. Yeah.
Quite lucky and a certificate to see that.
Yeah, that's right. Yeah.
The marketing stuff.
Yeah, marketing stuff, yeah. So yeah, we have a lot in common, Professor Botella. Yeah, that one thing. My message to you all that it's better because the technology moving very fast. So it's better to work technology based. Yeah. The platform based on the technology, not the commodity based, because then we can apply our technology to a broad spectrum of plants and not just plants, but it's also work for animal, for humans. And in the past, when I worked with for genetic diversity using long time ago before the DNA-based marker, we work with isozyme. So I also work with animal. So like a mouse or rats in the forest. So we check the tail, yeah, a little bit of the tail, and then to see the movement and then the diversity. So technology is what you call it, species borderless. Yeah. Okay, one more question. Okay, one more questions. Okay, from here on site from Mbak Alfia, please. Maybe you can use the also so that the online participant can listen to you also very clearly. Yeah. Yeah.
Okay, Prof. Jimmy, thank you for your presentation. Very interesting. And I want to know, your method is can apply to diagnostic. And how about if we want to screening which genetic plant it can be applied in for screening, or if we want to technology to partially screening.
Yes, yes, absolutely. That can be applied to anything that you need amplification to determine. It can be applied.
Just one thing. Yeah, Pak Edi this time. Yeah.
Because gene sometimes need a specific primer for like this. The gene is inside the gene. So how work is this stick for detect this primary from gene?
Well, you need to know what you are trying to detect. Okay, so this would allow you to do it, but you need to know what are you trying to detect, and you need to know the conditions for amplification. What my technologies will allow you to do it is in like 30 seconds instead of 4 hours. I know because see, they're sitting next to you. In the workshop that we have in Laos, they spend something like 8 hours purifying DNA.
Yes.
And then they have a coffee break, and in the 20 minutes that they have for the coffee break, they did exactly the same with my technology. And then after the coffee break, they kept working because they still didn't know the results. So, what they did in 8 hours, they did it in 20 minutes during a coffee break. That's the difference.
Heidi, further question?
Yes, sometimes I need to detect a gene. Is need a specific primer. So the sometimes I want to detect my GM plant, especially for potato, for resistant to Phytophthora infestans. Yes, I need a specific primer for to amplify fragment this gene. But this dipstick is look like snow. How to work? Okay.
So what happened with your GN lines is you're using a bacterium, I guess,
Yeah, mostly.
Alright, so you have a TDNA there. You can choose whatever, like you're using MPC in the system, you're using Canvas in the system. You would have your plans already developed for that. Okay. But again, instead of you can do analysis of 20 different lines, and it will take you, the preparation of the samples to the PCR for 20 lines will take you something like 10 minutes with this, instead of 8 hours and 20 kits to do it. Now, the PCR parameters that you're using depends on your TDNA. So most of the time, people will analyze the resistance. That is the most common thing to analyze to check whether they are transgenic or not. All right. Other times, depending on what transgene is it that you're putting, or you're using a 35S Pro model, you can use primers for the 35S. It's your pick which primers do you use. What the method would allow you is to get 20 plants and go number 1, number 2, number 3, number 4, number 5, follow the machine, press the button, you're finished. We do use it for transgenics a lot because it's very simple.
Yeah. So it's make our life easier.
Yeah.
Yeah. So I think next week we're going to try dipstick, right? Yeah, because we already prepared only 2 solution buffer needed. The Edward solution buffer and then the wash buffer, and we already prepared. So anyone want to have a look when we work next week using dipstick on Tuesday, maybe? Yeah, because yeah.
You need to pay.
Yeah, that's right. Oh, thank you very much. You remind me. So you are on my side. Okay. Yeah, so you are all welcome. I mean, it can be not just for plants people, but also for animal and human. Yeah.
As long as we understand that we are trying to optimize better.
Yeah. Yeah, yeah, this time, yeah. Yeah, at least you can have a look how it, I mean, the procedure, whether it's going to work or not, because we're going to try.
Okay,
Yeah.
maybe next week we will try potato transgenic.
Okay, you can try, yeah. So any other, yeah.
Different number, different different fragments.
She has calculated this, so she knows it.
Yeah,
Okay,
I mean, yeah, for the potato, yeah.
Yeah.
So we can try potato, a transgenic potato, and then also we can try also for the fusarium of banana and other crops, all different crops we can try, and then it's worked for all other crops, then we can publish together. Yeah, so the procedure works for different spectrum of crops at Brim. Yeah. Yeah, so in our research center. So yeah, we can try all different crops. Yeah. I mean, for the first time, of course, it depends on the crop itself. We need to optimize each of the crops and the procedure.
Depending on the crops.
Yeah, yeah. Yeah, that's right. Yeah.
As you said, it's developing under PPP.
Yeah.
because of
For the phenolic compound, yeah. Yeah.
It's a little bit of play that you need to.
Yeah.
My sample is a potato in vitro.
Yeah.
So maybe it's easy
So
to
it's not really.
Yeah, easier.
It's from
Yeah.
Yeah.
So we can try a potato, cassava, and banana. Okay. Yeah. Next week. Yeah. And sugarcane, of course. Of course, that's our main task. Yeah, with sugarcane. Okay.
Maybe I have a phone number and then one is control. Just potato. No, not here. And that is
Yeah.
yeah.
Sugarcane transgenic, yeah, transgenic sugarcane. She's the one that working on sugarcane also. It's in my group.
I gotta find the young one. Because I grow this in the garden.
You grow transgenic sugarcane in your house? No. No.
Standar.
Yeah. Okay, so sorry, it's very lively here on site. Yeah.
Let's start before we have 50 chairs.
Yeah. No, okay. Yes, it's very lively in here. I mean, those who join online used to come here with us, join us. Well, it's 2:45, and normally, Jimmy, from 1 to 3 is actually normally two speakers. But today, for you, only one speaker. So you got privilege. Yeah. Special, yeah. So, okay then. So thank you very much, all of you that all already participated in this FS3 lecture series of Research Center for Genetic Engineering of Green. You are all still, I mean, we are welcome for you to come to work with us and then to see our work in the lab next week. Yeah. So thank you again, Jimmy, Prof. Jimmy Botella. And then, yeah, as I said, this is the first time we are collaborated and the first time with Indonesia. So it will be in your CV, it will be one country added. Not just all the countries and then the ASEAN countries, but it will be Indonesia there.